Hello बच्चों, welcome to Hello Biology. This is the 9th lecture of chapter Principles of Inheritance and Variations.
Today we are going to discuss another exception of Mendel inheritance, that is, multiple allelism.
More than two alternative forms of a gene occupying the same locus on a chromosome, or its homologue, are known as multiple alleles.
Students, this image shows the concept of multiple alleles, where a single gene exists in more than two alternative forms in a population. Human ABO blood groups, eye color in Drosophila, and coat color in rabbits are classic examples of multiple allelism. Remember, although many alleles exist in a population, an individual can possess only two alleles at a time. Have you ever wondered how a single gene can give rise to many different alleles? Multiple alleles are formed due to repeated mutations occurring in the same gene over time. These mutations take place in germ cells, so they are called germinal variations. Such variations are often meristic in nature, meaning they affect the number or arrangement of body parts. Therefore, repeated mutations increase genetic diversity and lead to the formation of multiple alleles. Now, students, let's discuss the characteristics of multiple alleles. All multiple alleles are mutant forms derived from the same wild allele. They are always present at the same gene locus on homologous chromosomes. These alleles control different alternatives of a single trait. Depending on their interaction, they may show dominance, codominance, or incomplete dominance. Students, now let's understand the entire concept of multiple allelism with the help of the ABO blood group system. First, remember that a single gene, called the I gene, controls blood groups in humans, but this gene has three alleles, IA, IB, and I. Since more than two alleles of the same gene exist in the population, this phenomenon is called multiple allelism. Now, all these alleles originated from the same ancestral or wild allele through mutations and are present at the same gene locus on homologous chromosomes. Although three alleles exist in the population, any one individual can inherit only two alleles because humans are diploid organisms. These three alleles produce four different blood groups. IA gives blood group A, IB gives blood group B, IA and IB together produce blood group AB, and II produces blood group O. Here, IA and IB are codominant to each other, while both are dominant over I. So the ABO blood group system is the best example of multiple allelism because one gene exists in more than two forms, occupies the same locus, and produces different alternatives of the same trait, blood group. Many alleles can exist in a population, but an individual can possess only two alleles at a time. So students, after understanding the ABO blood group system, let's look at another classic example of multiple allelism, eye color in the fruit fly, Drosophila. In Drosophila, eye color is controlled by nearly 15 different alleles of the same gene, making it an excellent example of multiple alleles. The normal or wild-type eye color is red and is represented by the allele W. Due to repeated mutations, several new alleles have evolved, producing different eye color variations. Among all these alleles, the allele W produces white eye color and is recessive to all other alleles. Therefore, just like the ABO blood group system, Drosophila also demonstrates how a single gene can exist in many alternative forms and produce different phenotypes in a population. Students, till now we have discussed multiple alleles. Now let's move to another important concept in genetics called lethal genes.
So let's move ahead on lethal gene.
Have you ever wondered whether a gene can be so harmful that it causes the death of an organism? The answer is yes. Such genes are known as lethal genes because their expression may result in the death of the individual carrying them. Now look at this diagram. A lethal allele is an allele that has the potential to cause the death of an organism. However, death does not always occur immediately. The time of death depends on when the product encoded by that gene becomes essential for survival. If that product is required during embryonic development, death may occur before birth. If it becomes important later, death may occur during childhood or even adulthood. Therefore, lethal genes can affect organisms at different stages of life, ranging from early development to adulthood. The key point to remember is that a lethal gene is not defined by when it causes death, but by its ability to ultimately reduce viability and lead to death under certain genetic conditions. Students, now that we understand what lethal genes are, let's look at some important examples found in nature. One classic example is the lethal gene controlling coat color in mice, where certain homozygous combinations lead to death before birth. Another example is Huntington's chorea in humans, a dominant lethal disorder that usually appears during adulthood. The Manx cat is also a famous example, where the gene responsible for the tailless condition becomes lethal in the homo. These examples show that lethal genes can affect organisms at different stages of life and modify the expected Mendelian inheritance patterns. If you found this lecture helpful, don't forget to like, share, comment, and subscribe to Hello Biology for more easy-to-understand biology lectures. Thank you, and see you in the next class.